Hello students, we'll start with the second semester syllabus which deals with the enzymology and cellular metabolism, and this video gives an introduction to enzymes. This slide explains a simple enzymatic reaction where enzyme reacts with the substrate to form a transition state called enzyme substrate complex, and this enzyme substrate complex undergo a process to release the product and the enzymes are regained back. This explained a simple enzymatic reaction where enzymes act as a catalyst, where these enzymes they act as a catalyst in the living organisms. We call them as biocatalyst. Coming to the definition of enzymes, the enzymes are proteins functioning as catalysts that speed up the reaction by lowering the activation energy. Activation energy is the energy required by the substrate molecule to collide with the enzyme. The enzyme catalysts regulate the structure and function of the cells and organisms. Enzymes are biocatalysts that speed up the reactions of digestion and also metabolism. They are located in the cells, cytoplasm, mitochondria, tissues and body fluids. There are two types of enzymes, endoenzymes and exoenzymes. Endoenzymes that function within the cells, and most of the enzymes are of this type. Mostly the metabolic enzymes. Example is cytochrome oxidase. Exoenzymes, enzymes that are liberated by the cells and catalyze the reaction outside the cell. Example, digestive enzymes, they are released from the digestive glands, that is amylase, lipase and protease. The exoenzymes which are outside the cell shown here, and the endoenzymes which are within the cell. The substrate molecule where it reacts with the enzyme to release the product. What are the characteristics of enzymes? Enzymes are globular proteins, increase the rate of reaction without being used up, do not affect the properties of end product. Enzymes are reusable, catalysis reaction rate in very small amount, and they are very specific. One enzyme catalyzes only one typical reaction, and sensitive to pH, temperature and substrate concentration, etc. Some enzymes they need a cofactor for proper functioning. A fundamental task of protein is to act as enzymes, catalysts that increase the rate of virtuality of all chemical reaction within the cells. Even though most of the enzymes are proteins, some RNA are also capable of catalyzing some reaction, and most biological reactions are catalyzed by proteins. In the absence of enzymatic catalysis, most biochemical reactions are so slow that they would not occur under mild conditions of temperature and pressure that are compatible with life. Enzymes accelerate the rates of such reactions by well over 1 million-fold. So reactions that would take a year in the absence of catalyst can occur in a fractions of seconds if catalyzed by appropriate enzymes. Cells contain thousands of different enzymes, and their activities determine which of the many possible chemical reactions actually takes place within the cell. This slide explains the energy diagram for catalyzed reaction and uncatalyzed reaction. The reaction illustrated is a simple conversion of substrate into product, because the final energy state of P is lower than that of the substrate, and the reaction proceeds from left to right, and the activation energy is very less for catalyzed reaction and it is more for uncatalyzed reaction. Coming to the history of the enzymes, in the early of 70s, that is 1700, the early 1800, the digestion of meat by stomach reactions and the conversion of starch to sugar by plant extract and saliva were known. However, the mechanism by which this occurred had not been identified. And we have to remember the contribution of Louis Pasteur and the German physiologist Wilhelm Kuhne and Edward Büchner for their contribution. In 1850s, Louis Pasteur presented a theory that sugar is converted into ethanol by yeast, by ferments, and he recognized that ferments were acting as catalyst in fermentation. The word enzyme was coined by German physiologist Wilhelm Kuhne in 1877, and the enzyme literally means in yeast. In 1897, Edward Büchner discovered that fermentation of the sugar was possible with the enzyme zymase, and he received the Nobel Prize for the chemistry in 1907. So when we are dealing with the enzymes, we have to know what is the region where specifically the substrate binds. This region we call it as active site. The area of the enzyme where the substrate bind is called an active site. The structure has a unique geometric shape and is a crevices that is designed to fit a specific molecular shape of a substrate, and that region is called as an active site. Here the chymotrypsin is shown, and the region marked here, it is the active site where specifically the substrate binds. Active sites contain residues that bind the substrate and also participate in catalysis. Active sites sometimes contain a cofactor, and the active site residues have several important properties, such as they have the charges, they have a dissociation constant, and pK, that is called as pKa, and hydrophobicity and flexibility. What is the chemical nature of enzymes? Enzymes may be simple proteins, and also they are complex with other non-protein parts also. Simple protein enzymes, they contain simple proteins only. Example, urease, amylase and peptide are the etc. And complex protein enzymes, they contain conjugated proteins, that they have the protein part called apoenzyme and the non-protein part called prosthetic group associated with a protein unit. These two parts constitute what is known as holoenzyme. Holoenzymes, the term refers to active enzyme with its non-protein component. The term apoenzyme is an inactive enzyme without its non-protein part. If a non-protein part is metals such as zinc, iron, it is called cofactor. If the non-protein part is a small organic molecule, it is referred as coenzyme. Same thing I have given in the flow chart, where holoenzyme, it consists of a cofactor and apoenzyme protein part. The cofactor, it may be prosthetic group or coenzyme, and prosthetic group, that is, they are usually small inorganic molecules and they are usually tightly bound to the apoenzyme. Coenzymes, they are organic molecules, they are loosely bound to the apoenzyme, and some enzymes are proteinish, simple proteins. Same thing, the difference between the prosthetic group and the coenzyme or cofactor. Prosthetic group, non-protein molecule, again coenzyme, cofactor, also non-protein molecule. Their prosthetic group is tightly bound, coenzyme, cofactor, loosely bound. They have the stable incorporation in case of prosthetic group and dissociable in coenzyme, and prosthetic group cannot be dissociated, coenzyme separable by dialysis. Holoenzyme, the enzymes with protein and non-protein components. See the diagram here, it is a holoenzyme, you have a cofactor and coenzyme, and it is a catalyst, that is the active site. Complete catalytically active form an enzyme consists of apoenzyme, inactive form, and its cofactor. This is the difference between active site and the binding site. So active site I have explained you where specifically the substrate binds. The region where a protein, RNA or DNA to ligand bind to a protein, that region we call it as a binding site. So active site, they exist in enzymes. Binding site, they exist in all the biomolecules like proteins, RNA, DNA. The function of active site, as we know, that is responsible for reduction of activation energy and the chemical reaction, thereby increases the reaction rate. Binding site, it is responsible for binding of specific substrate or ligand with the molecule. Again, this is a picture which shows the binding site and the active site, catalytic site and the binding pocket, where the binding of the ligand or DNA, RNA which takes place. Okay, so I'll play a video for you. Okay. Just a minute. Just a minute.
Human body is a product of different chemical Human body is a product of different chemical reactions and processes. But what Human body is a product of different chemical reactions and processes. But what controls these reactions? In 1833, a French chemist, Enzymology, was the first to discover the vital force that drove these reactions and named it enzyme. Enzymes are substances, proteins, or in some cases, ribonucleic acid, RNA, that speed up a biochemical reaction by modifying specific substances called substrates. Enzymes are supremely selective in who they bind to and modify, hence their specificity. Mode of action. Easier said, but how does a tiny enzyme speed up a chemical reaction? The answer to this is simple. Enzymes perform this critical task by lowering a reaction's activation energy, that is, the amount of energy needed for the reaction to begin. Enzymes bind to their substrates, hold them in such that chemical bond-breaking and bond-forming processes take place more easily. Enzymes have a specific place in them called active site, where the substrate binds and real-time action takes place. Active site has a specific size, shape, and chemical behavior rendered to it by specific arrangement of amino acids. Thanks to these amino acids, an enzyme's active site is unique only to a particular substrate. In addition to the active site, many enzymes consist of a non-protein part called cofactor. Cofactors may be cations, positively charged metal ions, activators, bound temporarily to the active site to activate the enzyme, organic molecules, vitamins or vitamin products, coenzymes, that join enzyme substrate complex temporarily, aesthetic groups, permanently enzyme bound. Many enzymes only perform their catalytic role when associated a coenzyme. An inactivated enzyme, protein, along with its coenzyme, non-protein, makes up a system called Ola Enzyme. To add to the complexity, scientists term this inactivated enzyme as apoenzyme. And therefore, the equation becomes Ola enzyme equals apoenzyme plus coenzyme.
Okay, thank you for watching.